Hi everyone, and welcome to our summary on carbohydrates. This is the second part in our carbohydrate series, and this video is going to focus on the disaccharides. In terms of our specification, what we're looking at is this little statement here: the synthesis and breakdown of a disaccharide by the formation and breakage of glycosidic bonds. And this is going to include 3 disaccharides: sucrose, lactose and maltose. Before we delve into how we make these disaccharides, it's worth taking a moment to consider a phrase that's going to be associated with them. And this ties into one of those assessed practical tasks that you should have done, obviously, as part of your practical component. The phrase we're going to consider is reducing sugar. Now, one key rule to remember, if you like, is that all monosaccharides are reducing sugars. So if it's a monosaccharide, then we will have a positive reaction with the Benedict's reagent. If we consider our disaccharides, remember there are 3 we need to know about. Both maltose and lactose are reducing sugars, but sucrose is a non-reducing sugar. So what that means is we will have a positive reaction with Benedict's with maltose and lactose, but sucrose would give us a negative reaction if we just mix the 2 together. That's important as far as that assessed practical goes, which we will look at in a separate video. When we talk about reducing sugars, these are any types of sugar that can act as a reducing agent, hence the name, reducing sugar. And what that means is they're going to donate electrons to another molecule. In the case of our Benedict's tests, it's then donating the electrons to the Benedict's reagent. And the reason that they can do this is they all have this thing called a free aldehyde group. And this is what you can see over here. This part here is the aldehyde group, the carbon double bonded to an oxygen and then bonded through a single bond to a hydrogen. This little R here just stands for basically the rest of the molecule. So it could be any random bits of molecule attached there, but we don't really care because the only bit that we're concerned with in this instance is this aldehyde group, the carbon double bonded to the oxygen with a single bond to the hydrogen. What we're going to do then is we are going to have a little look at those 3 disaccharides in a touch more detail. One of the things you need to be able to do as far as those disaccharides go is you need to know what they are made from. There's no particular trick to this, it's just memorize it. So my suggestion here is to create yourselves a little flashcard with obviously maltose on one side and on the other side the 2 units it's made from, which in this case is 2 alpha glucose molecules. So as far as maltose, the disaccharide, goes, we've joined the 2 monosaccharides of alpha glucose together to make it. To make sucrose, it's an alpha glucose joined to a fructose, and then to make lactose, it's alpha glucose joined to beta galactose. The observant ones amongst you might have noticed that alpha glucose pops up all over the shop. So as far as our 3 disaccharides go, if they were to give you a question asking what the monosaccharides that make them all up are, and you're having a complete blank moment in your head, if you write down alpha glucose, then that's 4 of the 6 answers correct, because alpha glucose makes up 4 of those 6 units. The only different ones, we need to chuck a fructose in our sucrose one, and lactose has beta galactose. So realistically, if you really can't remember anything, write down alpha glucose and you're in with a really good chance of getting it right. Better if you know it, but if you can't, it's a way that you can have a decent educated guess. If you think back to one of the earliest videos we've done here, then we talked about an important reaction that was going to crop up time and time again. Here is the first time it crops up. We are talking about a condensation reaction. In order to join those two monosaccharides together, we are going to have a condensation reaction occurring. So if you remember, in a condensation reaction, we also generate a molecule of water, hence condensation. We need to remember the name of the bond that forms between those two monosaccharides when we form our disaccharide, and that is a glycosidic bond. So if you're looking for the name of the bond in our carbohydrates, glycosidic is the one you should be writing down in each case. So let's have a look and see how we actually make these disaccharides. At the top, I've literally just chucked in a couple of alpha glucose. How do we know it's an alpha glucose? Because on carbon 1, the hydroxyl group is below the ring in each case. So we have our two molecules of alpha glucose. Now what we're going to do is we're going to carry out a condensation reaction. So this is going to be our condensation reaction. And that means that we are going to form, first of all, a molecule of water. So remember, we're going to make a molecule of water as the end point. Now, in order to do this, we then need to join those two monosaccharides together. They can ask you to draw the link between them. So the way that we do this, and I'm going to just draw these out for you so you can actually see. We've obviously got our 5 carbons in the ring joined to the oxygen. Put in your CH2OH. Then we can just fill in the blanks, if you like, the hydroxyls and the hydrogens around the rest of the ring. So once we've got this, for our first one, we're only going to draw the one at the moment, then we're going to have a situation where something's going to happen, and I'll just change my pen colour, with this hydroxyl and this hydroxyl group. So we've got our two monomers and they need to join together. Now the condensation reaction means that we are going to be losing a molecule of water made of 2 hydrogens, 1 oxygen. So what's going to happen is from that carbon we are going to end up with just the oxygen. So the hydrogen is going off to be part of the water molecule. Then on the other side, what we've got, we obviously just quickly draw the little ring structure again just so we've got it in place. And then we've got hydrogen, hydroxyl, hydrogen, hydroxyl, CH2OH, hydrogen there. Didn't give myself enough space. Now, what we do have is, here we would have had our hydroxyl group, but in order to make water, if we've already lost the hydrogen from the alpha glucose on the left of our diagram, we then lose both the oxygen and the hydrogen from that hydroxyl group from the alpha on the right. That means that carbon now forms the bond through the oxygen. So what we have here, this part is our glycosidic bond. Now we can name these glycosidic bonds because it depends on which carbons they are actually attached to. So in this case, we always count our carbons from the right of the oxygen. So what we have is carbon 1, so that is carbon 1, and then on this molecule on the right, 1, 2, 3, 4, so that's carbon 4. So in this case, what we have is a 1,4 glycosidic bond. So 1,4 because the bond forms between carbon 1 of one molecule and carbon 4 of the other molecule. And that's it. That's how we draw it. If we then consider what happens when we've got a disaccharide and we're going to break it down into its individual monomers, then this is our hydrolysis reaction. the second reaction I told you was going to come up time and time again. The way the hydrolysis reaction works, remember hydro, water, lysis, splitting, we are going to basically split these molecules apart by adding water to them. As always, if there are bits that you've looked at in this video that you're still not 100% certain on, take the time now just to go back over it in a bit more detail. Use your textbooks, use revision guides. There's additional resources over on my website as well. Link is in the description box below. And of course, don't forget to subscribe so you can see when we add another video as well.